Okay, so we are going to talk about these two groups of fungi, and they are close relatives, and they are both our good friends and enemies. So that's why I call them cousins with double-edged sword. So first let's talk about what kind of family they are from. So both of ergot and epicloe, they are from a family called Claviceps. And this family is composed of members and kill caterpillars, kill caterpillars and spiders and nematode, I show you here three pictures, cordyceps and Turbiella heptocilium. And some of those species has already been developed as biocontrol agents, such as Bavaria and Metarhizium. They have been used for control all kinds of insect pests. And the last picture, you see the line and the leaves with some fungus, and those are the fungus called used to be called Ashersonia, and I worked on this fungus when I did my PhD. So that's why I put it here, and it used for control white flies and scale insects. However, the two cousins we are going to talk about today is not associated with bugs, but they are associated with plants. And on this tree, in that red box, you can see these two groups of fungi, they are closely related. And one thing common for them is they both had a bitter and a sweet interaction with a human being. So in next about 40 slides, I'm going to share with you a little bit of knowledge I know about them and about their relationships with human being and taxonomy overview and some genetic diversity in Canada. I will spend more time on ergot because I worked on ergot for about 4 years, and epicloe we just started not long ago. So for ergot, the picture here I show you I took in the field. It's an infected plant called broomgrass infected by ergot, and broomgrass is one of the most common hosts of ergot. So this is how the ergot make living in the field, in the nature. And those black greens, those are the resting body of the fungi. We call them sclerotium, or for plural is sclerotia. And in the fall, they will fall on the ground and over winter. Next spring, they will produce this little drumstick-like structure they call the stomata. And those are actually the sexual reproduction structure. And sexual reproducing propagules, we call ascospores, they arranged in this big head in a special order. And when time comes and those spores get mature, they will be dispersed first in the air and carried by wind, land on another flower, and the spores will germinate and penetrate the flower, travel all the way down to the bottom of the ovary, and then they come back, penetrate to this ovary, and keep growing, keep producing a lot of asexual spores we call conidia. Conidia were wrapped in very sticky honeydew. In a very sticky honeydew, and those honeydew will ooze out. And you can see this picture of the flower with some like honey drops like that. And those are actually conidia. And the insect will land on it, stick on it, and when they fly to another plant, and they will transfer the spore on another plant, get reinfected. So this will play again and again during the season and get more and more plant infected. So then for the infected flower or ovary, the the fungus will grow in it and keep grow, and the mycelia in the end and before the end of the season, they will get hardened and become dark and produce this structure we call resting body. And this resting body will overwinter and the cycle will come back again.
How much of the plant is colonized by the mycelium?
Sorry, say that again.
How much of the plant is colonized by the mycelium?
How much? It's only the
That little bit.
part,
Cool.
not the whole body. Only the seed, and one plant can several flowers get infected and produce multiple sclerotia,
Uh-huh.
multiple ergot. Did I answer your question?
Yeah, beautifully. And are each sclerota, are they a different individual or are they all connected?
That's a very good question, actually. That's a very, very good question. And so there is a chance one plant can be infected by different fungus, and they are all ergot, but they are different identity actually.
Yeah,
There is
cool.
and different genotype. We had the experience found on one plant there are two different fungus actually.
Yeah.
That's a very good question. So can I carry on?
Oh, yeah.
So it is this black sclerotia. They contain over 200 toxic compounds, and those compounds also classified to different classes. And those compounds can be toxic and can be useful. Those compounds make them double-edged sword. So the dark side of the ergot is if human being or animal, if you accidentally ingest a large amount of ergot contaminated with in the grain, or you take little but for a long time, it will also impact on your health and produce and can lead to kind of syndrome we call ergotism. It's a kind of toxicity called ergotism. Ergotism, there are two forms. One is a convulsive ergotism, another is a congruous. Convulsive is a nervous dysfunction and cause like trembling, shaking, twisting, confusion, even hallucination. And gangrenous is like a blood vessel constriction. And you see the picture here, and the hands is almost like burned out, and some people can lose their legs because of the lack of blood, and so you can lose your extremities. And both of those syndromes can cause death. So ergot human interaction presumably start from Mediterranean agriculture started, but the the documented epidemics is start from 857 in a place right now belong to Germany, and in other parts of the country, world, such as France, Russia, and Finland, and where the rye was planted as a staple food, they are all like different levels of epidemics, and cause different levels of mortality. And even modern times, there are also some cases. This book I show you on the right, the day of St. Anthony's fire, is recorded a case happened in France in 1950s. So it can still be a problem, and especially for animals. And the tragedy is not only the mortality they cause, but also the political persecutions because of their mysterious uh, cause of this toxicity. And the most notorious case, you might know the Salem witch trial. And it happened in Massachusetts in 1600s. And during the whole process, there are 200 people were persecuted, accused for practicing witchcraft. And because of some victims get these convulsive symptoms, I thought because those people, witchcraft make these people get sick, get them got sick. Um, and some people some people was killed actually, for executed for this reason. And there are also similar kind of cases in Europe as well. It is very sad. And so there is a saying called set a thief to catch a thief. So bad things can be used to fight against bad things. And so this neural interaction function of ergot has been used for medical uses. In ancient time, folk medicines used them to help childbirth. Nowadays, actually, pharmaceutical companies have, based on those transmitters, developed all kinds of medicines to treat this list of disease. You can see this list, and you might identify some of them you hate the best. And the medicine I show you here, hydrogen, is prescribed for treatment of dementia, because by increased blood flow and the neural grow in your brain. And it's quite nice, right? And there are also some experiments confirmed this ergot alkaloid has anti-tumor and also antimicrobial functions. So for pharmaceutical companies, these are really a chest of treasures to, to look into. And ergot, beside this dark side and a bright side, and there are also some controversial side. So ergot, it was speculated that during the ancient mystic rites, people take the extractions from ergot to make the connection between human being and their gods, and because of this hallucination effect, right? And there are also some, throughout the history, there are some artists, philosophers, and some people take the extractions to look into different forms of consciousness. And so I think probably you know this figure who he is, and genius, but I think the whole thing is very controversial and just don't follow. And so in terms of ergot, there are a lot of stories about them. So I just feel like it's too much for me to wade in, and the more you read, the more story you find. But for me, I'm a taxonomist, so I'm more interested in their identities and classification. In terms of the identity of those black greens, actually there were all kinds of hypotheses in early days, such as they might be sun-baked grains, or excessive formation of sap, or the mutation of grains. And until 1771, botany, I think he's a botanist at that time, and Geoffroy recognized ergot as a separate plant because at that time fungus was still considered as plant, separate plant from their host. And Decandle is also another famous mycologist, and he agreed Geoffroy's observation and named the fungus, gave the fungus a name called Sclerotium clavus. And at times, people did sometimes see the little drumstick structure produced on the ergot, but people thought it was just another parasite on the ergot. Until Tulasson, 1853, he found it's not another fungus, it's a sexual stage of this fungus. So he renamed the fungus as Claviceps purpurea. And those on the right, the image is a hand draw from Tulasson. It's very beautiful. So nowadays, the species of Claviceps are described based on morphological characters and alkaloid content, sugar content, host range, and also molecular markers. For morphology characters such as the sclerotia, shape, color, size, on the left, you see the picture, and also the stromata, the color, shape, size, and surface of the big head of stromata, and also the length and the width of the stipe. For the asexual propagule conidia, there are two forms, macroconidia and microconidia, and they all come different shape and different sizes. So besides morphology, alkaloid content can be different. As I color-coded here, you can see different species, like purpurea, purple-colored, has this ergopeptide, ergotamine, and others are like different species have different set of ergot alkaloids. For host range, most of the species has very restricted host range, and they usually only infect one genus. But for some, they can infect several genera. However, for the species Claviceps purpurea, the one we care the most because it infects our crops, had a very wide host range. It can infect 500 or 400 species in 19 genera and in 3 subfamilies of the grasses. It's very wide. So which made people suspicious this species might actually be a complex of species, which means it has more species. It's not just one species, it's multiple species. And it turned out to be true. And people in not long ago, 2008 or 208 and 215, they use molecular marker and also phylogenetic analysis confirmed in the original pre-molecular concept of C. purpurea, there are 4 different species. Arudinis, as I show you, color different, like 4 different color. Those are 4 species in the original, considered one species, single species. And currently for the whole genus, there are 79 named species. And some of them, as we said, some of them only produce this honeydew stage. And the mycologists gave it a name called Sphaecella. Some of those fungus already connected with Claviceps. Some actually not Claviceps. They are different. They are not ergot. They are different, belong to different genera. And there are also 21 of those species has not connected with any of those either Claviceps or others. It may be or may not need further confirmation. So the most recent work by the Czech Republic group they separated the whole genus to 4 sections, which means the genus actually is very complex. And the picture here I show is a group, and I visited in March before the lockdown, Czech Republic. The guy stand there who is the leader of the group. So in Canada, we have a National Mycological Herbarium, DOM, where we get where host a lot of material, and we use it as a resource to do our research. And in that collection, there are 700 Claviceps, and 90% of them named as Claviceps purpurea. So at that point, we were thinking maybe, although there are sporadic other species, and we were thinking maybe partly because they are just not being seriously studied taxonomically by a taxonomist. So in 2016, we started doing some serious taxonomy, and we collected sample with the help of a lot of people, and in the end, you will see a lot of people help and collect a big collection of ergot from all around the country. And we amplified multiple genes. We use a molecular method and phylogenetic analysis, developed this tree. And on this tree, you can see 4 species recognized previously by Czech Republic. It's all recovered. And in addition to these 4 species, we found some new species. And this one, C. ripicola, was collected from the lake river banks or the sand dune, or it's kind of semi-wet habitat. And on Amorphila, this plant I show you here on the left is Amorphila, and it can also from Phalaris or Calama grostis, and it's actually quite common here in Canada. And this is another species I collected from Newfoundland. Newfoundland is from marshland, and on Agrostis, this species is characterized by this colony feature, special colony feature on artificial media. Other species, they are all fluffy, and this one is so compact, and it's like a root-like structure and compact. It's very unique. And then we have Quebecensis. Quebecensis was collected from Quebec. You can tell from the name, and it was also from sand dune and on Amorphila. This species is featured by the Canidia shape. It's oval shaped. It's different from other species. And the last one, C. occidentalis, is actually from our cultural collection. We have a Canadian Canadian collection of fungal culture, and it historically accumulated a lot of cultures, and we got it from there. Unfortunately, we don't have the original material anymore. Originally, it was collected in 1950s by a scientist. His name is Campbell from Alberta. So we only have the culture state. We don't have the ergot. And besides the morphology, in terms of pathogenicity on major crops, there are also a little bit of differentiation. And we inoculated the cultures on crops. And the last species, occidentalis, seems like much less virulent compare other 3 species. And also for the alkaloid content in sclerotia, besides occidentalis, for that one, we don't have ergot, and other 3 species all have a different combination of major and minor ergot alkaloids. So the results of this study has been published recently in Mycologia. So back to this graph, and you might ask what happened with this big clade, big cluster, Claviceps purpurea central stricto. Is there any kind of genetic subdivision or variation in it, or it's just all the same, and because of the sexual reproduction, and they all exchange genetic material, so they're basically the same. So that is a question we asked ourselves. We looked into more analysis suitable for population level and deal with the relationships, tease out the relationships and between organisms. There is a kind of a genetic interaction. And there are different approaches I showed you here: coalescence theory, statistic, and split, and also the softwares. We looked into these new analytical tools. And based on this new analytical tool, more deeper analysis, and we found actually in that C. purpurea clade, it can be separated into three genetic clusters. So I call them GC. GC genetic cluster stands for GC1, 2, 3. And GC1 is more distantly separated from another two. So next question would be, is this separation just accidentally or is consistent? And so in order to answer that question, we looked also other methods like a Bayesian approach and principal coordinate analysis. And you can see the graph intuitively, you can all see they support the hypothesis from split tree. So then our next question would be, if this genetic separation correlated with the geographic region or the host, because of different hosts separated them. So to answer that question, we looked the genotypes in all different genetic geographic origin. We looked, in order to simplify the question, we separated to three different regions: Canada West, Canada East, and US West. We did get some samples from US West, from Wyoming and Colorado and Montana from that place. So, but from this graph, you can clearly see in all these places and all these three GCs exist. So which means there isn't a correlation between geographic separation versus genetic separation. So our next question is whether they are correlated with the host. So, and among the hosts, we also separated them as three groups. Crops include barley. Hodum is barley. Sickle is rye. Triticum wheat, and triticale is a hybrid between triticum, between wheat and rye. And also triticale node crops, elements, and a few genera, and non-crop triticale, and also brumi. Remember, we have brumus is the most common host. And among these three groups, you can also tell all the group has all the three genetic clusters. So it's not really separated by the host. But so the mystery is, what is the cause of the separation, of genetic separation? So that's our next question. Our research right now is doing, and it could be some like meeting type or some kind of stuff, but it's all just a hypothesis. So now the take-home message for ergot, our study, yes, that's a lot. So the take-home message for ergot is, in Canada, agriculture and the surrounding area, there are 8 species. and 4 of them are new, and we just described. And most common species is still Claviceps purpurea. However, in that species, there are also some genetic subdivision separated to 3 genetic groups. However, right now we don't know what is the cause of the genetic subdivision. I guess that's all I have to say about ergot. Now I'm going to switch gear, talk about choke
Before
fungi.
you move on, can I ask a question about ergot?
Yes, sure.
I'm I'm curious. You referred to it as a plant pathogen. Um, does the fungus hurt the plant, or does it just hurt the animals who eat the plant? Like, can the plant reproduce?
That is another very good question. It is the same person asked a very good question. Actually, you know, in the beginning, from our human perspective, it's pathogen, right? It hurts us.
Because it
It
hurts
hurts
us.
us. Actually, recently, there are some genetic analysis based on genome analysis and based on all kind of analysis. There is an indication that this fungus probably can increase the fitness of the plant.
Yeah.
And that is actually happening for choke fungi. And I'm going to talk about.
Oh, cool.
Yeah.
I have another question about the life cycle. Well, actually, about the honeydew. Is the honeydew created by the fungus or the plant?
By the fungus.
It is amazing.
Yes, yes, by the fungus. The fungus produce, grow mycelia and then produce asexual spores and wrapped with this, you know, they absorb nutrition from the plant, right? So probably from some moisture from the plant, but it is produced by the fungus.
That's amazing.
Yeah.
Thanks.
Okay, good. So now we talk about the choke fungi. Choke fungi is a little bit different from ergot, but they have something in common, something different. What different is these fungus actually grow inside of a plant. We call them endophytes. 90% of the time, they just stay inside of the plant and they absorb nutrition from the whole body, the whole plant body, and absorb nutrition and keep the plant healthy. And then when the plant produce seeds, they penetrate to the seeds. So then they pass on next generation along with the seed. When the plant seeds harvested go to another place and they go together as a vehicle. So that's an asexual stage. And 90% of these fungus grow like this. And occasionally the fungus will pop out from the plant and produce a structure as I show on the right side of this cycle, the bottom, and produce this structure called the stromata. And the stromata is a sexual producing structure. This stromata usually produced just underneath the fluorescence of the grass. And they absorb the nutrition and grab the nutrition, prevent the plant produce seeds. It's like a choke in the throat. So that's why it's called choke fungi. Choke fungi. And they produce this stromata. This stromata will, inside of this stromata, there are sexual propagules and they can eject on the surface propagules, and the propagules can be carried by also by another insect, by a kind of beetle. Beetles, they carry it, go to another plant, and also actually on another stromata, and they had a special pattern of very interesting behavior, dancing on the stromata, and make actually by doing this help the fungus mating mating, and then produce new propagules, infect another plant. So, but majority of the fungus is still asexual stage. And these fungus, similar to ergot, they actually produce more alkaloids. Besides the ergot alkaloids, they produce also another 3 classes of alkaloids, and they can be good and bad. That's the same. That's the same story with ergot. So the bad thing is the most common host for Claviceps. Oh no, not Claviceps. Epichloi. For epichloi is like a festuca, lolium, and those forage grasses. And when animal eats them, and will get toxic, and produce a kind of trigger, a kind of a symptom look like ergotism. So I will show you this video.
Oh my God.
Yeah.
It's called the ryegrass stagger?
Yeah. It's called the ryegrass stagger. That's right.
Oh my God.
The disease is called the rye. And it's very, very sad. And yeah, so it can be very harmful for animals, for our stocks. And so there is also another grass in northwest China. It's called the drunken horse grass. And when the riders ride horse, they pass that area, and some of their horses probably eat this grass, and they will fall asleep for a couple of days.
For a couple days?
That's why, yes, a couple of days, and that's why people call it a drunk horse grass. In the beginning, they thought it's a plant, but actually it's not the plant, it's the endophytes inside of the plant. And the human toxin, it also happened, and as recorded, wheat can mixed with darnel seeds, right? Darnel seed basically is, I think it's lolium, or I'm not quite sure which species is it, but it's a common host of endophytes. So the seeds contains this endophyte, and with the toxic, when the wheat mixed with it, and it's toxic to human being. But the bright side, so the bright side of epichloi is the grasses, to answer your question just now. So the grasses with endophytes, they can like defer the insects. Insects hate those grasses. So in a way, it's kind of protected the grass. And this actually has been used for control some insect pests on grasses, on turf grasses, and also on other grasses, as a pest control, pesticides, as a biopesticides, without using real pesticides. It's kind of environmental friendly approach. And also for the grasses, of course, sometimes it's not grass, it's crop. And if they have endophytes inside of them, they have increased fitness, so they can endure more drought or the bad nutrition. It's nice that then you don't need to use fertilizer, and can also help to develop a resilient agriculture to face the climate change.
Mhm.
And then there is also another story I just learned actually not long ago. It's there is a kind of grass called sleepy grass. In some culture, they use this sleepy grass to treatment of insomnia, and it's also in folk medicines. It's a kind of useful of this endophytes. And those endophytes had been studied by, in Europe, in US, and based on those studies, they are quite common actually also on various grasses, and both exist in seeds and the plant themselves. This is just a snapshot of all a lot of researches. So taxonomy, because of the structure you can see, and there, oh, I show you the stromata. They're really not much morphological character. In the beginning, it's just a yellow cased on the grass. So all the species on cool season grasses were all called Epicloid typhina. That's all just one species. And in 1990s, there are three groups of uh, researchers led by these three big figures. I call them three musketeers. Musketeers. And so they did a detailed morphological and biological study and mating experiments, and found that on cool season grasses, there are 10 species. They recognized the 10 species. And among those 10 species, only 4 of them exist in North America. And those species are presumably uh, they can't mate between each other, presumably, biologically. And also morphologically, there are very minute morphological differences they used, such as the density of stromata. You see on the top, this photo, the parathecia, each one of those structure called parathecia. The parathecia is like separated from each other, where the bottom one, they are all kind of merged together, and those can be a character to differentiate the species. And also the ascospores, which are produced inside of that parathecia, they can be uh, whether they separate to small pieces or they are just keep a one long thread like that is a character. And also the shape of the part spores was also used for separate species.
Mhm.
In 2001, Shadow Lab did more work on use molecular markers and did phylogenetic analysis. And it turns out that all the phylogenetic traits are incongruent with each other, which indicate there are actually some interaction between those biological species. And this figure I showed you here is it's like a assembling of 1,000 phylogenetic tree and developed by Beast. And the analytical tool I showed you before is better for the group with genetic interactions. And on this tree, you can tell Epicloid typhina on the top, Epicloid typhina and Epicloid sylvatica, there are some kind of gene exchange. You see the reticulate relationship. And also at the bottom, among these three species, there are also some gene exchange. So which means the biological species, not all the biological, some is good, but not all of the biological species are congruent with phylogenetic species. And so they are, but why and nobody knows. And you see the bottom, amaryllis is a North American species. How come amaryllis had an interaction, gene exchange with European species, and nobody knows why? So in our herbarium, again, there are 56 specimens. They are all called Epicloityphina, and collected from 7 provinces and on various grasses. So now I have a question for the audience. And so look like we have a much less specimens compared to ergot. So what could be the reason why we had so much less samples? But you don't have to answer now. We can all come back after the talk, and maybe you can also put on the chat, and we can come back to this question again. And so a student in my lab, Cassandra Bison, she was from University of Ottawa, and she did her honor project. She's kind of interested in the fungus and try to figure out how many species actually we have, and even though they are all called just Epicloityphina, and she developed molecular data and to grind the fungus and extract DNA, amplification, multiple gene, and sequence, and then combine her data with Craven data, the tree I showed you earlier, and we generated this tree. And all the samples with red lines are the samples she developed. And on this tree, you can tell actually there is only one single sample that was actually collected by Jonathan. Jonathan, he's in the room now, and he collected last year. That was actually the only one true Epicloityphina, and all others are actually different species. And we got some elemi and amaryllis, and also glaciaria. We had a good collection of glaciaria, new collection in Ottawa area and east and west, so which allowed us to had a better photograph to describe the species. And this is what we did, and it's going to publish soon to recharacterize the species. And during this course, we actually found that the original type was, when you describe a new species, you use types, and those types, actually, there are some problem. We made corrections, and with our nomenclature expert, Dr. Scott Redhead in our center, and we really appreciate her expertise. So I think that is all what I have to say about those two groups. And now these are the major contributor to all the data we developed. On the top is Parvash Shohoki. She is my technician, is very indispensable help to me. And we have volunteers and co-op students worked in our lab at different times, and then they learn the techniques, but meanwhile, they also develop the data for move our research forward. And besides that, we have a lot of colleagues and also collections and people, organization, every can, and they helped us from one way or another. And also like bioinformaticians and molecular and taxonomy and all kinds of people. And so I think I'm not going to read all the names. But without their help, we definitely cannot make any progress. With that, I thank you for your attention. And it is a precious weekend evening. And thank you for fun with me.
Thank you. That was fascinating.
Thank you. So should I stop sharing?
Stop share so we can all see each other.
Okay. Can you see me actually?
I think it's still transitioning. Oh, we can still, there we
Okay.
go.
So I stopped the sharing. Okay, so back to the question, back to my
Uh-huh.
question. Back to my
I
question, why we have so few specimens of epicloe compared with ergot?
Oh, so there's
What
more
could be the
ergot?
reason?
Oh, I had it backwards. I was thinking
700,
about,
700 ergot, but only 56 epicloe.
is it because it's asexual most of the time?
I will give you 100.
Oh,
That's what I thought. That's also I kind of promote, it's because they are mostly invisible, invisible. That's why I think we need work on them. And we are trying to put a proposal and use a molecular marker to detect the ones in grass, but they are invisible.
yeah.
I didn't even realize that I had only Tiffina from around, so I'm kind of happy. But yeah, Epicloe Tiffina is quite pretty if you manage to find it.
Find
Interesting.
it.
Oh, wait. Yes, very interesting. We got so many samples. They are all just different species, and only the single one you got, and only half stomata, only half stomata, and unfortunately, uh, in that half stomata, half of them probably was used to develop a culture. So right now we only have 1/4 stomata left of a single specimen. So next time, Jonathan, when you go to, where did you collect it? I think it's on the east side. And
Yeah, it was in Rockcliffe Park, and I remember quite exactly the spot. It's quite easy to go, but I didn't went this year, unfortunately. Next year.
I see. Yeah, I see. Yes, I understand. I think the host is Dactylis. I think I suspect because you only have a small inflorescence. I think it's Dactylis. And watch on Dactylis. But anyway, thank you for thank Jonathan, and he collected a lot of samples for us, and also not only epicloe but also powdery mildew, a lot of ergot and a lot of samples.
And I found some more powdery mildew this year on plums.
Okay,
Cool
On
I
to
plum?
see.
show.
Yeah,
I have a question.
yeah.
How do regulatory authorities keep ergot and epicloe out of the food system?
Oh, yeah.
How the regulatory what? I'm sorry. Can you repeat the question because you're
How do we keep ergot and epicloe out of the food system?
Mm-hmm.
Yes, that's also good. I think for ergot is, we are all recognized in Canada, and we all know the problem, which is good. We know the problem, which is good. And then in the organization like a Green Commission and everywhere, and there are standards. There are standards. And in that standard, yes, I actually had the last slide. I didn't show you how do we control ergot disease, how to manage ergot disease. So there are like three big category uh, measures to control the disease. And the last one, let's from the last to the first. The last one is after you harvest, you got the ergot, right? And some, such as the Green Commission, they will screen the crop and use some kind of special equipment to shake it and take away all the ergot. That's the mechanically take away the ergot. And for the certified seeds, there is a standard, each for let me see what's the standard for
So
certified species. For
the crop
certified
isn't lost. Sorry.
seeds, such as for wheat, barley, and oat, you can only allow 2 sclerotia on 1 kilometer crop, grain. So for rye because it's more heavy, more susceptible, so you can allow 4 sclerotia body. So there is a regulatory criteria mechanism to.
Is it very common? Is this something that a farmer would have would come in contact to or with often?
Yeah, it is very common. It is very common, but so good thing is we don't have a lot of rye. We don't produce although we do produce rye, but after all, rye is not our stable food, right?
Yeah.
But rye is most susceptible. However, on wheat, oat, and barley, they are all susceptible. And each year there will be a certain amount of sclerotia developed in the crops. And the Green Commission, they will screen the crop and the grains and take away the sclerotia from the crop to make them safe, and certify the grains. And other than take the sclerotia away, there are also some other method to control the disease, such as deep plow. After you deep plow, and the sclerotia might be plowed buried deeper, right?
Yeah.
Buried deeper, and then the spores cannot disperse.
Yeah.
That's one. And another way is to clean the field heads, because some other, like broom grass, and the cordgrass, they are all the hosts. If you cut them out, take them away, sanitize, and they are less inoculum and to infect the crops. And also there are also some variety is more susceptible, some variety is more resistant. So they are all different practices to control ergot. And also another information is in Canada, we are not allowed to use pesticide to control ergot because they are not safe. However, for epicloe, it will be another story because they are not visible, right? They're invisible in the grasses. And I don't see a lot of study in Canada actually about the presence of epicloe. And I see Keith is in the audience. He probably know more information historically, whether there are some study in Canada studied this group of fungi. But he's mute. Oh, he's mute. Can I unmute him? Oh, I can. Can't unmute him anymore.
He's gonna keep it a secret.
Yeah, keep it secret.
Or it's a different Keith.
Oh, maybe.
Yeah, maybe.
Maybe.
Maybe I can ask a question then, since we were
Yes,
just
Tom.
pesticides. I'm curious to know if the different species of Claviceps, and for that matter, epicloe, do they attract different species of insects for dispersal, or is it kind of generalist? I know it was beetles for epicloe, if I remember right.
For epicloe, I read more on epicloe, and yes, different species. So because different epicloe, they probably because of the scent or they evaporate a kind of chemistry and can attract different species. And the genus called Bountiful beetle, and there are different species of Bountiful. They are related with different epicloe species. However, for ergot, I don't know, and I only know for ergot. I didn't read through my readings. I didn't see any information. But one thing I found very interesting is when I collect ergot and when the ergot get late of the season, when the ergot late of the season, the ergot will get empty, like a hollow inside, and you will see a lot of little beetles hover on it. And I'm kind of curious, so I caught some of those and gave it to him, our entomologist. So, and he recommended me some reading, and those beetles, I don't remember the genus name. It's called ergot beetle. It's called ergot beetle. But
Oh,
I haven't read a lot about them, whether they are species specific for the ergot transfer.
Okay. Yeah, it might be kind of interesting to look at that. We have a GCMS now in the lab.
Yeah.
So if you wanted to look at, we could do some headspace tests and see if they're releasing any volatile compounds that might be different
That
between
would
different
be,
species.
yeah, that would be very interesting. That
Yeah.
would be very interesting. And also, another thing is the beetles eat ergot. You see, they don't get toxic.
Huh.
They get sick.
Yeah.
Right? So probably they have some kind of mechanism to, you know.
Yeah. So were you saying that the epicloe has like an anti-feed effect when it's living as an endophyte? Like, do the insects not eat the grasses, or was it just larger animals that don't eat the grasses?
For some, you mean for epicloe, right? The
Yeah.
grasses with epicloe, some insects. Okay, your question is whether all the insects don't eat the grass or enter only specific insects.
Mm-hmm.
The question, right? Yes, I don't know actually. I only know in general, endophyte injected in infested grass can you know defer the insects.
Okay, I'll stop asking you questions about insects now because I know this is a fungus talk. Sorry about that.
That's okay. And also they are quite common use for golf grasses.
Huh.
Golf.
yeah, golf courses. And so I wonder if that would keep the deer away too. I mean, if we
Yes.
saw what it did to the cow
Yes.
and what it would do to other grazers. Or what about if someone was walking their dog? I guess dogs eat grass to throw up anyways, but
Epicloe, yes. They are not only defer insects, also animals. It's kind of their life strategy. You know, they
yeah,
want other people eat them, right?
they want to protect their host. It's
Yes.
amazing.
Yeah, herbivore, all the herbivores, not only insects, but also animals. Yeah, that's right. Well, more questions? What? Very nice to see all the faces.
Yeah. I thought it was curious. You started to tell us about correcting a type species in the collection, or I mean, not just in the collection, but in the world. I thought that once a specimen was named the type, that it was it, and you couldn't go back on it.
You're right. You're right. I didn't explain well. Yes, people actually it was by shadow, and when they designated the type, they made a big mistake. So we corrected it. It's not that we designate a new type. It's not.
Oh, you didn't change it.
It's still their type. They made mistake. They, you know, as I told you, the epichloi they can mate, right? Actually, he did a lot of mating tests, Chris Shadow, and he did a lot of mating tests, and he put all the different mating types and in one specimen, so we separated them. And also, by the way, I did my postdoc in Shadow's lab. So I did, it's very interesting. That was 10 years ago. Oh, 14 years ago. 14 years ago. 14 years ago, I worked in Shadow's lab, and I work on Claviceps, but not ergot. I work on the toxin part, the molecular part. Work on the molecular part and gene level. So then I got an immigration paper ready in Canada. So Shadow's lab is in Kentucky, University Kentucky. So I moved to Canada, and at that moment, and one of my classmates, and I said, oh, I feel so bad. I only come here only a couple of months, 8 months, and now I'm leaving. And he said, I'm sure I can feel in some years you will come back again, study again. And it is actually true. After 14 years, and I come back to study ergot again. And people, mycologists had a saying said, the fungus, once the fungus find you and they won't let you go,
Mm-mm.
keep you.
I believe it.